Good morning, ladies and gentlemen. Thank you so much for joining us today. I think it's kind of timely that we had that huge crescendo of music right at the end there, given that it's a pretty diabolical issue that we're collecting together today to discuss, and ever an evolving space with particular to COVID-19. So I would like to take the opportunity to thank our incredible panelists for coming together today. Just giving an opportunity for a few more people to jump on the line. We've had some tremendous interest in this subject, as you can understand. And we're absolutely delighted that we have the opportunity to share with you today how we have been really agile with some existing research programs, but also established new research programs in the face of the global challenge that is facing us all. To begin with, I would first like to do an acknowledgement. On behalf of the University of Queensland, I would like to acknowledge the traditional owners and the custodianship on the lands of which we all virtually gather today. We pay our respects to their ancestors and their descendants. We continue cultural and spiritual connections to country. We recognise their valuable contributions to the Australian and global society. Well, with that, I would like to welcome our director, Professor Ian Henderson, Dr. Larissa Labson. We also have Dr. Mark Blaskovich with us and Dr. Loic Yengo. So this is our last of the Meet the Researchers for this year. So we're leaving it on a significant note. Originally, we had intended to cover off on 4 different vantages of the ways that our researchers are approaching the health and ageing. But in face of what we have seen across 2020, we thought, what better time to showcase to you how our researchers here are harnessing their fundamental efforts to complement the work of the vaccine development with looking at ways and mechanisms and new therapies which will address what has happened amongst the population now that COVID is in community. So in today's webinar, we'll feature 4 different unique vantages, but what I think is very cool about the gathering of researchers that we have with us here today is actually how integrated some of their approaches are, and it'll become evident as they share more with you shortly. Particularly, I know Larissa and Mark are working in partnership. I know that Ian and Mark are working in partnership, and the project that Loic will share with you very shortly is in fact one that can wrap around all of these different approaches and feeds into it. So it's a fantastic example of how IMB, as Australia's leading life sciences research institute, harnesses our integrative model. So we put fantastic people in the same building, in the same space together, and then we let them put those minds together and have at some of these big issues, just like the one that we are discussing today. And so before we jumped online today, I did try to look for a couple of stats of this and that. But as you can understand from what the researchers will share with you shortly, it's an ever evolving space. So as we are lifting restrictions here in Queensland today, South Australia is looking at how they need to readdress an emerging threat again. And then, of course, we are in a completely different landscape here in Australia and even New Zealand than our friends, family and loved ones are experiencing overseas in different territories. And we do just want to note, we recognise that there are those facing big challenges, even as we are here gathering online today. So without much more ado, it's my delight to first pass over the microphone duties to Dr. Larissa Labson. So Larissa joined us in the latter half of 2019. She is our NHMRC CJ Martin Early Career Fellow, and she's an immunologist who studies our immune system and how it fights viral infections. As you can understand, it's been a very busy year for Larissa. She's been very much a go-to in the UQ specialist space in being able to comment on the discoveries that have been discussed globally, and she's been on all different channels in international forums to actually share her knowledge. Larissa uses immunological, genetic and imaging techniques to understand how influenza and SARS-CoV-2 interact. So she'll share with you, that's actually behind COVID-19. with our innate immune cells and trigger inflammation. Her research aims to uncover new treatments for acute viral infections, just like COVID-19, and to develop better vaccines and antivirals. With that, I pass over to Larissa.
Hi, Kamara. Thanks. Marlene, can you share the slides for me? I think you've got them, right? I will talk nevertheless. So as Kamara mentioned, I'm an immunologist, and I'm particularly interested in understanding how our immune system recognizes when we've got a viral infection and how it decides to respond. So we all talk about COVID-19 constantly, and I just wanted to refresh with you all what actually COVID-19 stands for. So COVID-19 refers to the disease that is caused by infection with the virus, which is SARS coronavirus 2. So it's called a coronavirus, and in the picture on the right here, you can see that they've sort of highlighted it in yellow. You can see the viral particles, and they have this crown-like appearance, hence the name corona. And what's on the outside is this spike protein. And you might have heard a lot about the coronavirus spike protein. Basically, it's what all the vaccines are, essentially. They're this spike protein that we're delivering into the body in some particular way so that our immune system can recognize that spike protein and mount an effective response against it. So people can be infected with SARS-CoV-2 and it doesn't necessarily lead to disease. So that's an asymptomatic infection. But of course, as we know from COVID, and from this year, actually, for a lot of people, if they do catch SARS-CoV-2, then they get severely sick. And that disease is COVID-19. So Marlene, if we can go to the next slide. So basically, I like to think of how sick we get in terms of how our immune system responds to the virus. So we really see a couple of different outcomes. The first, which is on the left there, is that we manage to eliminate the virus. So we clear the infection, we're resistant, and we feel fine and we feel healthy, and everything is great. The second scenario is that we don't manage to clear the virus, but we kind of come to a balance with the virus. So it's persistently there, but it's not killing us. Actually, it's just sort of surviving with us. And the best example of that are bats, which happen to be viral reservoirs. So actually SARS-CoV-2, we think, came from a bat originally. And bats have this unique feature in that they have all these viruses, but they don't get sick from having all these viruses. And that's kind of this tolerance is actually sort of one avenue that people are thinking about when it comes to viral infection. Instead of trying to clear the virus, can we just, live with the virus essentially and not get so sick? And the final scenario, which is what we see with severe COVID-19, is that we become susceptible. And what happens there is either we don't clear the pathogen, so we don't clear the virus, it takes over and it basically uses up all our nutrients and blocks our normal function, meaning that we succumb to disease. Or on the other hand, in its attempts to control the virus, the immune response actually gets out of control. And so we have all this collateral damage to our tissues and not just the viral infected tissues, meaning that we actually get really sick from what the immune system has done. So what we really want to achieve is on the left, the sort of just right approach where we're able to eliminate the pathogen. And probably the best way to do that is when our immune response is working the best itself. And so we think of that as when we've cleared an infection, we then become immune to that virus or pathogen. And this is because our immune system is pretty smart and it likes to keep a memory of all the things we've been infected with before, because that means it's able to respond much more quickly the second time around. And that's basically the entire point of vaccines. What vaccines are trying to do is trying to trick your immune system into thinking that it's seen the virus before, so it knows how to respond best. But obviously for vaccines to work and for us to have long-lasting immunity, even from natural infection, we need to have an immune response that is just right. On the other hand, if our immune response is too little, and so the virus is overrunning, the kind of approaches we could take would be to use antiviral drugs. So to use drugs that are able to stop the virus and stop it replicating. So I think a good analogy for that is like antibacterial drugs that target bacteria. And on the other hand, and this is something that my work particularly focuses on, is can we use anti-inflammatory drugs or immune suppressors to dampen down that immune response when it's gotten out of control? So Marlene, if you can go to the next slide again, please. So if we're looking at COVID-19 in particular, we think that it actually sort of manifests itself in a number of different phases. So you might have heard people talk about this, but really, the ultimate time for getting a vaccine is before we get infected. And then if we've managed to be infected, but, it's still really early days and maybe we're just starting to feel a bit of a sniffle, or we know we've been exposed, but we don't feel sick at all. This is actually the viral phase, and any symptoms that we have in that first week is probably attributed to what the virus is doing in the body. And actually that's the best time to treat with those antiviral drugs, because that's when the virus is at its peak. And the same for those monoclonal antibodies. So recently, Eli Lilly and the Regeneron ones, which Trump was treated with, they're best administered in that viral phase, because we know that sort of after about 7 days of being infected, actually the levels of virus start to decrease. And so if you're starting to be hospitalized in that second week of infection, actually treating the virus is not that beneficial anymore, because the virus isn't really around that much. And it's actually the immune system that's causing all the damage. So you've probably heard of this cytokine storm. And again, that's something that we're particularly interested in. So that's basically this huge inflammatory response. And we know that in this phase, actually, one of the treatments that's shown the most promise is dexamethasone, which is an immune suppressant. And so that means that actually when people are presenting in hospital with COVID, treating with anti-inflammatory drugs might be the best way to actually save people's lives. And then we get into the coagulation phase. So I think you've probably heard about some of the cardiovascular effects that are going on. And this means that basically your blood is starting to thicken, and there's all this vascular dysfunction, meaning that you're at higher risk of stroke and heart disease. And then we also know that there's a late hyperinflammatory phase, which we really still don't understand, but we think this is what's happening with the so-called long haulers, the people who just aren't managing to get back to baseline levels, back to original fitness after they've been infected. So in addition to focusing on the cytokine storm phase, and this is something that I'm doing together with Professor Kate Schroeder, who's a world leader in inflammation research here at the IMB. And Kate's my official, I'm in Kate's lab, and she's a great mentor. And we're really investigating this together. And then we're also investigating the coagulation phase with a couple of fantastic vascular biologists we have here at the IMB. And that's Dr. Emma Gordon and Anna Lagendijk. So we're really trying to understand how is the virus or the virus-induced inflammation triggering this endothelial dysfunction. And we do all of this together with Dr. Kirsty Short over at SEMB, who's a leading virologist. And I'm sure you've probably seen Kirsty on TV or heard her on the radio as well. So what we're also interested in at the IMB, as I mentioned, we're interested in all these things. So we have a fantastic selection of chemists and peptide biologists. That means we have unique access to a whole heap of compounds. So we could actually look for antiviral drugs as well as anti-inflammatory drugs and anticoagulant drugs, all within the same space. And we can harness our research to look at all of these different phases. But to do that, we need to work with the live virus. And that's actually why I'm a bit harried this morning, is because I just finished working with the virus. So Marlene, if you could go to the next slide, please. So I just wanted to show you this picture. So this is cells that we've infected with SARS coronavirus 2. And what you can see in red is one of the viral proteins. So in blue is the cell's nucleus. In yellow is the outline of the cell. It's the cell's cytoskeleton, so its structure, basically. And yeah, what you can see is that many of these cells are infected. And so we used microscopy like this to determine which cells and where in the cells they're being infected. And we also use other methods to see if these cells are triggering an inflammatory response and releasing these pro-inflammatory cytokines, which is basically how inflammation is perpetuated. So yeah, as I mentioned, what my particular research is focused on is how is inflammation triggered in COVID-19 and other viral infections, and so how can we stop it? And as I sort of just summed up there, we're interested in how the virus and inflammation affects our blood system. And if Marlene, if you can go to my final slide. So this is just to show you what it actually looks like when I'm working with SARS coronavirus 2. So we do this in PC3 laboratories. These are special contained labs that have negative pressure so that we prevent any potential escape of the virus. And we are decked out, as you can see, in full PPE. And this facility is currently over at SCMB, which is in a different building. But at the IMB, we actually have a building and our own PC3, and this is really going to make a huge difference for us being able to really push this research forward. So I just wanted to show you that working with the virus itself is no trivial matter. I'm actually the only person at the IMB at the moment who's currently trained to work with SARS coronavirus 2. And that's why I had to run there this morning for an experiment and then quickly come back here. So with that, I'll hand back to Kamira.
Thank you very much, Larissa. And I'm so glad that you pointed out at the end there the idiosyncrasies around the PC3 laboratories. It's a good time to mention that we are tremendously grateful for the support of both John and Georgina's story just in this year. They are wonderful friends to the organisation and also lifetime members of our directors circle. And given their ongoing interest in the work that's been happening in this building for many years now, we were in a position that we could sit down with them and let them know about some of the challenges that we faced from an equipment perspective. And they have got behind us in supporting our capital requirements to upgrade those facilities. And they've also dedicated a little bit more money to our ambitions to tap into the biodiversity that is around Queensland to search out new potential microbes to support future therapies for all manner of different issues. Larissa, that was great. And no doubt there's 101 questions that are going to come out of what you've just shared with us. So this is a good opportunity to let everybody know if you do have a question, please pop it down in the Q&A section. But we will also be coming back to each of our panel members at the end to check in with them on any specific questions that you might want to ask that they can answer within the breadth of their expertise. There may be some questions that are not apparent. So handing the microphone duties over now to our Dr. Mark Blaskovich. So I'm delighted to share that recently Mark stepped into a position as a group leader here at the IMB. And he is also the director of our Centre for Superbug Solutions here. Mark is an antibiotic hunter and medicinal chemist with 15 years of industrial drug development experience prior to stepping into an academic career. Mark has been developing new antibiotics to treat drug-resistant pathogens and using modified antibiotics to detect bacterial infections. He is a co-founder of the Community for Open Antimicrobial Drug Discovery, or COAD for those of us who can't wrap our tongues around that, a global antibiotic discovery initiative, and has led a number of UK industry collaborations focused on antibiotic development. Today, Mark will present on improving the existing method of testing wastewater that, despite the value and sophistication, still has significant shortcomings. When developed, this test can detect infections in larger populations much earlier than is currently apparent. Thank you, Mark. Over to you.
You're muted, Mark.
Yep, I got you. Thanks for the introduction, Kamira. So as the introduction indicated, our research has in the past predominantly been focused on discovering solutions to bacterial infections. But there are a number of different areas of research where we think we can repurpose some of the research that we're currently doing and apply it to trying to find solutions to COVID and viral infections. And I think one of the key things to point out is that we our research group doesn't have any direct experience working with virus. So the collaboration with Larissa in particular is very important to enable any of this research to be conducted. And so we're waiting anxiously for her to get the PC3 lab up and running within IMB so we can help to advance some of the research projects that we're thinking of doing. So I'm going to actually present 3 different research projects. So the first one would be talking about the wastewater solutions. And if you can, am I sharing my presentation, Marlon, or are you putting it up? Because I don't have an ability to share my screen. All right, so if you can just flick through about the next 4. So there's been a lot of publicity recently in the press talking about the ability to do surveillance for SARS-CoV-2 in a larger population. So the advantage of this technique is you're not relying on testing individual people to see if SARS-2 is present in the community. And it's particularly relevant to places like Australia, where you're hoping that you actually have no SARS-2 in the community at all anymore. In a larger population where it's quite prevalent, you're obviously going to be finding lots of traces of the virus in the sewage that's been shed from people who have been infected. But for monitoring a community where you don't think you have any, but want to be able to pick up that perhaps there is a undetected number of cases in the community, the ability to detect the virus in the wastewater is very important to track and monitor that. And so here's some recent publicity where, for example, it was detected in Southeast Queensland and found in Cairns. And the way they do this is that when people are infected, they shed the virus, and in particular, the DNA components that make up the virus that encode for the virus, fragments of those are shed into the sewage and survive intact. And so if you sample the sewage, you can use techniques that amplify and allow you to detect these specific viral sequences and show that people have been infected in the community. However, this is detecting just fragments of the virus. It's not actually detecting live virus. And so what we're proposing to try doing is seeing if we can develop a similar technique, which is actually able to capture intact virus rather than just fragments of virus, and then use that to look at, do you have any live viral cases in the community, rather than people who may have been infected in the past and are still shedding dead virus fragments. And so for this, we're looking at trying to repurpose a technique that we've developed for capturing bacteria and determining whether there are small quantities of bacteria present in large volumes of water. So if you could go to the next slide, please, Marlon. Sorry, this just gives an indication of how detection wastewater works. So you have your population of people, their virus is getting shed when they're using the bathroom, goes into the wastewater system, samples are collected at the wastewater facility, you put that in the lab, extract out the DNA, amplify the DNA, and are able to detect that potentially there is virus present. Just go to the next click. And this detection is being done even within UQ. This paper was published a couple months ago, including a couple of UQ collaborators. And this is based on this detection of the fragments of the DNA from the virus. And next slide. And the cool thing about this technique is that you can actually track back and localize exactly where that infection has occurred. So using the sewage network, you start with the wastewater treatment plant, which is a little box off in the right there. And then you can go back tracking through the sewage system and sample at different parts of the system and track back to a specific suburb and location to see where that infection may actually be localized. Next slide, please. So what we're hoping to do is try to repurpose our antibacterial to try to potentially capture and detect live virus. And we don't know whether this is feasible yet. We think it might provide some advantages over just detecting the fragments of the virus, but that has yet to be shown. And it has been shown that live virus can survive in sewage for 1 or 2 days. So potentially it does give you this ability to detect at an early stage whether there are live infections going on in the community. So the concept is that we've developed for detecting bacterial infections is you take very small magnetic nanoparticles, and then you functionalize the surface of these magnetic nanoparticles with something that's able to capture the bacteria. And so similarly, we just changed the functionality that we're using and changed it to something which is able to specifically trap the complete intact virus. Next slide, please. So we put that in a large solution of water. So a wastewater sample, like might be, a liter of water. By trapping the virus, we can then use a magnet to pull down these tiny magnetic nanoparticles that have the virus embedded with them. Next click, please. And then we're able to then transfer that into a very small volume of solution where we can now use a variety of different potential techniques to identify that there actually is virus present. And what we'd be looking at is looking at extraction of the RNA or DNA and then doing whole genome sequencing on that fragment we've taken out of the intact virus. And so this gives us the ability then to not only look at that whether virus is present, but also determine its sequence, so we can try figuring out whether the virus, if you have multiple types of virus present, or whether it's coming from a single infection source. So this is at a very early nascent stage where we've basically got the concept. We're kind of waiting for Larissa to be able to do some more testing with live virus to see if we're able to capture the virus and then detect whether it's still there, or use genetic sequencing to identify the virus. So the second technique that we're looking at potentially repurposing is an assay that we've developed that gives very high sensitivity to be able to detect components produced by the body in response to infections. So when you are infected by a virus, as Larissa indicated, your body mounts an immune response, and one of the components that it generates are antibodies that neutralize the virus. And there are actually a couple of different types of antibodies that are generated. And so what we're proposing to do is develop an assay that's able to measure these different types of antibody responses at the same time. And potentially that gives you a much better picture of what's going on in the body in terms of its response to the viral infection. And we don't know in terms of the multiple types of antibody responses, which ones are more important. And in particular for developing assays to be able to look at whether people are infected, looking at this immune response might give better sensitivity than detecting the virus itself in terms of whether they have an active infection. So this assay is what's called a sandwich assay. And so you take a sample of biological fluid that potentially has the human antibody response to the virus present in it. Next click, please. And we, again, are using magnetic nanoparticles. And these nanoparticles, next click, are functionalized on the surface with a component of the virus, which is the target of the human antibodies. And so this allows us now to trap the human antibody from the solution. Next slide, please. And then we add in a fluorescent nanoparticle, which has a anti-antibody. So it's an antibody which is able to selectively bind to the human antibody. And so now you've got this sandwich where you've got your target antibody sandwiched between the magnetic nanoparticle that captured it and a fluorescent nanoparticle which is able to light up the complex that you've now formed with a magnetic nanoparticle. And you can do the same thing using a magnetic nanoparticle that's selective for the other type of antibody and use a different color nanoparticle. So now you've got what's called a multiplexed assay where you're able to look at two different components using the two different colors of the nanoparticles. And the way that we then detect it is put into an instrument called a flow cytometer, where basically you're flowing a solution of these nanoparticles and captured complexes through a laser light. And the laser has several different components that it can detect. So the backscatter or reflection of the light off the particles can indicate the size of the particles that are passing through the laser, whereas the fluorescence from the fluorescent nanoparticles gives you an indication of which particles that go through have that fluorescent nanoparticle attached to them. And you can then separate out the different components. So some of the particles that are going through are just the magnetic nanoparticles with nothing attached. Some of them are just the fluorescent particles that just give a fluorescent signal. But some of them are these complexes, which are now conglomerates because you've got multiple components on them, where you've got a mixture of the nanoparticle and the fluorescent nanoparticle. And this conglomeration gives you much greater sensitivity and fits into a different, because you're looking at both types of laser detection. It gives you much better sensitivity over background noise. And so ideally, we'd be able to develop this and be able to detect multiple components at the same time with very high sensitivity and very good ability to look at low levels. And then finally, the last slide, going back to what Larissa was mentioning about trying to come up with new antiviral compounds, we have an initiative in terms of developing new antibiotics called COAD, the Community for Open Antimicrobial Drug Discovery, where we've essentially been trying to crowdsource new antibiotics. And we've been doing this by offering free testing for activity against a number of different bacteria to research groups around the world. And we've been doing this for about 5 years now. And with funding from UQ and the Wellcome Trust, it's been able to support us to offer this service for free. And it's been highly successful in terms of over 300 different research groups from around the world have been sending us compounds and over 300,000 compounds to date over the last 5 years to test for antibacterial activity. And so there's the opportunity to try doing exactly the same thing for antiviral compounds. So one thing, we could go back and look at a lot of the compounds we've already received and put them through Larissa's assay once she's developed a higher throughput version of it to try seeing if any of these compounds we already possess do have antiviral activity. But we could also go out to the greater global community and specifically ask for people that are trying to develop new antiviral compounds and don't have access to testing, particularly against live virus facilities. We'd be able to collaborate and do this testing for them. And basically, that's what we've got. So 3 very nascent assays or projects, a couple of assays, one for developing new antiviral compounds or potentially even immune modulating compounds. But as I mentioned, they're all very reliant on collaboration with Larissa and access to this PC3 lab that we're anxiously waiting for.
Thank you very much for that, Mark. There is a fabulous amount of information there. And I saw that you had particularly some questions coming through while that was underway. I think you've answered a couple of them, but I just want to assure our guests we'll return to those very shortly. But I think it's important that we keep moving because we try to squeeze many faces into today's event so that we can let you meet multiple researchers. And our next one is also another new group leader to the IMB space. It's my delight to welcome Dr. Loic Yengo. So he's a statistical geneticist with our program of complex traits genomics group. So that's he works very closely with Professor Naomi Ray and Professor Peter Vischer, who, for our repeat offenders joining us at these events, you're well aware of their work also. So Yengo joined us at the university in 2016 with a research background in applied mathematics, statistics, and molecular epidemiology. His current research focuses on understanding the genetic and phenotypic consequences of non-random mating, inbreeding and assortative mating in human populations, and on developing analytical methods for genome-wide association studies, or the GWAS. His presentation today answers one of COVID-19's biggest questions. Why are some people who contract COVID-19 barely affected while others developing life-threatening complications? This is a tremendously important piece of work that Loic is doing in partnership with those in his group. And I'm delighted to hand the microphone over to Loic to share more with us now.
Oh, thanks, Kamara. So I'll be sharing my screen right away if I can. Just trying again. Perfect. Right. So, well, I'll be showcasing the power of big data analysis and how that can be, if implemented quickly enough and at a large enough scale, deliver some important insights into a world-threatening pandemic, for example. So, as Kamara was saying, our main question is how do DNA variants influence symptoms response, in particular disease severity? And so that research question has been motivated by the observation that infection from SARS-CoV-2 virus manifests in a wide range of symptoms, with the vast majority of people being virtually asymptomatic, some people having sort of mild symptoms such as the loss of smell, loss of taste, but in few cases, unfortunately, also that leads to quite severe outcomes and death in particular. So there has been prior evidence for other infectious disease that DNA variants in the host, so in our case humans, can modulate well, not only the susceptibility but also the symptoms response and severity. And so this is the sort of the very question we're trying to answer. So my group has been looking into genetic basis of various disease in the past, and one of the successful experimental design we've been using is called genome-wide association study, and that experimental design is in essence pretty simple. We want essentially to contrast. We have a group of people with the disease and a group of people without the disease. And what we do is that we look at the DNA sequence of those participants, and in particular, we look for part of their DNA sequence where there are variations, and we look for overrepresentation or underrepresentation of certain DNA motifs between people with the disease and people without the disease. What we've learned over the years is that that type of experimental design is powerful. It can deliver quite important and deep insights, in particular when facing a disease for which we don't have much prior knowledge. But also, we've learned that the type of experimental design requires a large sample sizes. And so that inherently has led us to work together and to build consortium to study those questions. So early this year, my group have joined this international effort called the COVID-19 Host Genetics Initiative, which is a tremendous effort. So I just highlighted here the first, I'll say the early leaders of that efforts, but we've been joined by others in our groups, included. And so this effort at the moment comprises about 36 studies from 16 countries. It's talking about more than 300 researchers that are active every week. So we have this Slack channel, gathering many people from different backgrounds, trying to do as much as possible to collect data and combine data to explore those questions. So the latest update we have from that effort is that we've been able to collect about more than 31,000 COVID-19 positive participants out of about 1.8 million individuals. And I would invite you to have a look at this website where we essentially post our results at each iteration. So we've opted for this very open access model where pretty much every month we share our results and doubts and interpretations and getting all the feedbacks from the entire community. I also want to highlight, and you probably heard my accent, that we're trying to reach a wide community of people and not just English-speaking countries. So you can find content in up to 13 different languages, French included. So we've been quite fortunate in Australia not to have many cases of COVID-19 compared to other parts of the world. But it's still interesting and important for us to build the capacity to study COVID-19 at in particular potential subsequent waves and which can be of a different magnitude. And just wanted to highlight over and above that international effort, also in a quite important national effort led by my colleague, Professor Naomi Ray. So that effort will be is soon to be launched, and it will provide unprecedented data for people who have been infected, essentially trying to get their blood samples in a longitudinal fashion in order to understand, answer a lot of questions, but one of them is what are the long-term consequences of being infected with SARS-CoV-2. So this is just a screenshot from this web portal, which is soon to be launched. So over the well, I'll probably say about, it's operational since about 4 months. So what are the things that we found? Well, we started with the initial goal of understanding the whole spectrum of symptoms. And what we've found actually is that most of the information that, the genetic information, the genetic signal that we can detect comes from contrasting people with hospitalized patients versus people with mild or asymptomatic manifestation. And so there is a reason to that. And one of the reasons is also based on our strategy. We've tried to aggregate as much data as much as different sources. And that has in particular led to aggregate data from more reliable sources like hospitals, but also direct-to-consumer a platform like 23andMe, and people have been mostly self-reporting their symptoms, and that comes with quite a bit of heterogeneity, which can explain the reason why our signals are diminished. But nonetheless, we've been able to identify robustly 5 genetic regions where the DNA sequence show variation that is associated with increased risk of hospitalization. And just for those who are interested in gene, you may have probably in the audience, I'm assuming that some of you may have their favorite genes, but just highlighted here a few of those genes. And the reason also why I highlight those genes is because they play an important role in immune response, and some of them are cytokines, which loops back to some of the things we can work on with Mark and Larissa, who introduced their research questions at the beginning. So there's still a long path to walk. So we already have evidence that this type of experimental design can deliver new insights, and we keep on collecting data. And this Australian study that my colleague is launching will contribute to that. So we're definitely going to make more discoveries. But there's still a number of challenges, and that one of them is that of integration, in particular, to be able to bridge those variation that we detect in the DNA sequence with actual biology, with the immune response and gene expression. For some of those genes, we start to have a clear pattern about what exactly that DNA variant is doing and how it is changing the gene expression, the protein. So we're getting at some sort of basic biology, but we still a lot of things to learn on the go. And another important challenge is also methodological, because it's important to praise how responsive our different national and international research communities have been. But I think it's also a challenge that we're trying to get as much data as possible, and I tried to touch on that earlier. But that comes with the price of ascertainment. We have one of the early observations that we had about the risk of COVID-19 was that certain people with certain ancestry background were more susceptible. And so we have now evidence that it is partially true, but the early signals that we were seeing mostly came from ascertainment, where ancestry also correlated with socioeconomic status, and those more susceptible populations showed an increased risk, but that was nothing to do with their genetic background or ancestries. So this comes with a lot of challenges that hopefully we will be able to solve in order to implement this type of experiment quick enough, in the future if another pandemic hits us. Unfortunately, I think others will come. Sorry, I didn't want to end up on this very sad note, but yeah, it's still a long path to walk.
Thank you, Loic. I'm conscious that you've left it on that note, but I think it's probably a nice linking through to our next speaker, Professor Ian Henderson, and is also, I think, notated by Mark's presentation as well, in that already we were very firmly in the fight against superbugs. It was known that the next pandemic was coming. So that fight has not stopped in any way, shape or form. And any of the specialists in this field do allude to the fact that beyond this pandemic, there is the next the next wave beyond that. So with that, we'll move to our fourth and final speaker for this morning. And very much in line with what Loic was just sharing with us, one of the confronting realizations for particularly clinicians in this space in the early days of the pandemic was the antibodies produced the fight of the infection were making patients much more ill than others. And so this really intriguing piece of research that Ian is going to share with you today is such a novel and innovative approach to this very big issue at hand. But just to share with you a little bit about our director of the Institute for Molecular Bioscience, Ian Henderson's research group studies bacteria and their interaction with humans and animals. Their research is focused on the discovery of genes that are important to building the cell membrane of bacteria. Using genetic, biochemical, and structural techniques, they study the activity, regulation, and function of these genes and their products. Using mice as a model, they assess if the products of these genes are important for infectious disease. The group's purpose is to apply this understanding to the diagnosis, prevention, and treatment of infections that can lead to life-threatening diseases in humans and animals. Ian seeks targets for developing new antibiotics, vaccines, and therapies. This morning, he will introduce plasmapheresis, or what is much easier to say, the vampire therapy, and how it can save lives. Over to you, Ian.
Thanks, Kamira. And thank you for putting this together, and Marlon, and to Larissa and Loic and Mark for some fantastic presentations. So I think, Marlon, am I sharing my slides or have you got them? You got them, okay. So, you know, like all the other speakers alluded to, you know, the thing that we're most interested in as researchers is actually why do people die from COVID and what can we actually do about it? So these are the graphs, and I'm sure you're all familiar with looking these graphs up quite frequently, but these are the graphs from yesterday of the daily new cases of COVID globally and the number of deaths. And you can see where, you know, we peaked this week at 670,000 new cases a day across the globe. And of course, if you look at the graph on the right-hand side where there's the number of daily deaths, we've just exceeded about 10,000 deaths a day. And so the question then is why do you have all these people who are getting infected with COVID and recover, and you have these people who get infected with COVID and die? And what's the reason for that gap? And if we can understand that, then we can design some intervention strategies that prevent those people from dying. Now, as Loic alluded to in his talk, you know, the way we do that is actually comparing those people who get infected and survive with those people who get infected and pass away. So, Marlon, if I could have the next slide, please. Now, I just wanted to put this slide up here just to explain about death rates, okay? So the black line that you can see running across the top of the graph there is actually, so this is the data from the CDC in the USA. So the black line is the number of deaths that occur across the year, you know, the number of expected deaths that occur across the year. And that line actually hasn't shifted pretty much over the last several years. It's pretty much the same number of people dying in any given week across the whole year. Except for this year, you can see this blip on the graph. There's a big peak here, and that's the beginning of around the middle of March, or beginning of April, and that's the beginning of the COVID crisis in the US, and you have this peak of deaths. And then it goes down, and you've got the second peak here, and we don't have on this graph the third peak that is now coming. Well, what you can see from this graph is a couple of things, is that this is what the graph is graphing actually excess deaths. It's graphing deaths above normal. And so while the US reports about 200,000 COVID death cases, the actual number of people who have died more than expected is 300,000. And so we can actually that indicator suggests that the death rate due to COVID is actually higher than is being called through monitoring of infections. And that's also true in the UK and around Europe. So those numbers are about, indicate that we're about sort of 30% off on the number of deaths attributed to COVID. Now, the numbers on the right-hand side might be of interest, because we all think of COVID being a disease that really affects old people. And it is true that most of the people who have succumbed to the disease have been elderly. But in actual fact, you would expect the most number of people to be elderly, because that's actually where the most number of people, die. Most of the people who die tend to be elderly. And if you look at, and as you would think that if you looked at young people, you'd expect very few people to die in that younger category, under 25. And if you look across the age categories, what you're actually seeing is that people who are under 25 during COVID are actually 2% less likely to die, okay? And that may be something to do with the fact that people are in lockdown, so they're not out indulging in risky behavior. But you can see that the biggest risk category, and it's something that people probably don't appreciate, is actually 25 to 45, where you're 27% more likely to succumb to, or to die than you are if COVID was not around. Now, that's not big numbers. It's not big numbers because we don't expect people in that age category to pass away, but in actual fact, the risk is higher for that category than it is for others. Anyway, so the other thing you might notice from that graph is that the death rate is actually lower now than it was at the beginning. And so why is that? People have suggested that the virus becoming less virulent, etc. But in fact, it could probably be attributed to two things. The age profile. So at the outset of this, it was a lot of elderly people getting infected, and now there's younger people getting infected, so they are more resistant, if you want, to the infection itself, and to treatment. And so if you look at the very beginning of the outbreak, if you went into intensive care with COVID, the chances of you dying in intensive care were about 40%. And the chances of you dying if you were on ventilation were 70%, so 7 in 10. That number has come down significantly. So if you go into intensive care, it's now less than a 30% chance of dying. And if you were ventilated, it's about 35%. Okay, so it's halved. And so that's just about doctors learning how to deal with patients and to treat them and using different medicines. Okay, can I get the next slide, please, Marlon? Now, one of the things you may have seen is that children are relatively protected from this disease. Okay, but there have been a number of cases of children being diagnosed with a disease called or a disease like Kawasaki disease. So this is a multi-system inflammatory disease, and there's only a few children. But it was interesting that these children were infected with COVID, apparently recovered, and then 2 to 3 weeks later, you had this onset of this inflammatory disease. And this kind of like what the graphs that Larissa was showing at the very beginning, where you had those 4 stages of disease. But you can also look at just this graph here on the right-hand side, and when you plot the case numbers in blue against the deaths in red, you can see that there's a lag. The deaths lag, peak of deaths lag against the peak of cases. And that lag in deaths to cases, and also this onset of the Kawasaki disease in children 3 weeks after apparent recovery, suggested that the immune system must be driving some of this fatality. And so if we think about the immune system, there are really two branches to that immune system. There's what we call the innate system and the adaptive system. And the innate system is, think of an infection as an earthquake. It's terrible, everything shakes, and you're a building and you become unstable. The innate system are the engineers that are rushed in to stabilize that building to make sure it doesn't collapse. The adaptive system then is the bit that surveys the system. It's the memory that says we mustn't build buildings like that again in the future. And the main component of the adaptive system is antibody. Okay, so antibody, we can go to the next slide, please, Marlon. Antibodies are these little Y-shaped molecules, okay, and that the body produces in response to an infection. Okay, and those antibodies are like Velcro, they're sticky, and they stick to the surface of microbes, just like you can see here, and sticks to specific bits on the surface of the microbes. And these microbes then get targeted for killing by the rest of the immune system. And so not all antibodies are the same. You get a variety of different ones, IgM, IgG, IgN. They have a specific pattern of development during the infectious process. But what researchers had noticed with the patients who had severe COVID was that that sort of dynamics of producing those antibodies was completely dysregulated. People weren't producing the normal antibodies at the normal time. And so we had this hypothesis that that antibody could be playing a role in actually driving disease. And that's important if you're trying to develop a vaccine, okay? Because antibodies, vaccines elicit antibodies. It's an artificial way of inducing them so that they can actually protect you against disease. So, question is there any evidence that antibodies actually do have a role in disease severity? And the answer is yes, at least for viruses. You know, we know that people who get secondary infections with dengue, so they have antibodies to the dengue virus, that those antibodies can actually make the disease more severe. And we ourselves had noticed in patients with HIV that that some patients can actually develop antibodies that make them much more susceptible to sepsis. And based on that work, we had started to look at patients with cystic fibrosis and bronchiectasis, and those patients get infected with a bacterium called Pseudomonas. And we found that those individuals some of those individuals produced antibodies that actually made the disease worse, that actually allowed the bacterium to actually induce severe infections. What we hypothesize is if we could remove those antibodies, that those patients would get better. And so this is, you'll see on the right-hand side, is a picture of a chap called Jordan, who's an adult with cystic fibrosis. He had a lung transplant. And after his lung transplant, his wounds got infected with Pseudomonas, and we found that he had these bad antibodies. And as Kamira talked about, we used a treatment we called the vampire therapy, where we literally sucked out all of his blood, washed it through a filter to remove all of his antibodies, and gave him back his blood. And he actually recovered from those multi-drug resistant infections. There were no antibiotics left to treat this chap, and he recovered and went home to his wife and continued a happy life. So we are hypothesizing that we could actually treat some COVID patients using a similar vampire therapy that we applied to Jordan. Okay. And then lastly, if we go to the next slide, Marlon. The thing that you can see on the left-hand side there rotating is actually the spike protein from the surface of the virus. Okay, that is the target at which all vaccines are directed at. And the problem with that target is you can see that there are three pieces. And those have to come together to get the appropriate immune response. And that's why making vaccines has been for COVID and its similar viruses have been difficult. And the UQ vaccine has actually worked to clamp those together so that they're in the right conformation to be recognized by the immune system. But of course, it's taken an awful long time to develop these vaccines, and we would like to have things that actually work much, much quicker. And so we've been working for a number of years with Novartis and then GSK on meningitis vaccines. And we'd like to take the learnings from that vaccine's work to actually try and develop new, much more rapid vaccines that can be rapidly deployed for the next outbreak. So I can't say too much more about that, but we'll leave it at that. Thank you very much for listening.
Thank you, Ian. I think that was a wonderful place to end the synopsis and bring together comprehensively what we were trying to showcase for our guests through today's presentations. Now, this is obviously a hot topic. Our Q&A has lit up. There's a lot in here from one Marlon Othman, though, so I'm assuming you've picked up a couple of questions off the chat. But if you'll roll your minds with me back to Larissa's presentation when we were talking about cytokine storms, Charlotte Wheelow was asking, of the immune targets, are you planning on using, what immune targets are you planning on using to find a solution to COVID-19, Larissa? That comes from Charlotte, but I'll also just put a caveat to Charlotte's question. I believe she might be a microbiology student here at UQ. And so she's also sent the other question, how do microbiology students get involved in your different projects and support what you're doing?
Okay, well, I'll try and answer the first question. So that's actually the $1 million question, Charlotte, is what do we target? And so there are a couple of different options. And what's currently in clinical trials now are cytokine inhibitors. So you might have heard of an IL-6 inhibitor, tocilizumab, as well as a couple of other immunosuppressants. So you can have inhibitors that will block the effects of these pro-inflammatory cytokines. And that's basically what tocilizumab is, and also JAK-STAT inhibitors. And they're showing some promise in the clinic, but the direct cytokine inhibitors are antibodies themselves, so they're quite expensive to make, and they can have broad spectrum effects. So what we'd really like to know, well, what is triggering the cytokines? So we're actually looking at what signaling pathways your immune system activates in order to release those cytokines. So we're looking at toll-like receptor signaling, inflammasome signaling, those kind of innate signaling pathways, because the idea is if we can stop the inflammation at the source, that might be the best way to go about it. And as for the having microbiology students, we actually have lots of programs throughout the year. So there are summer semesters programs, but also undergraduate programs. So feel free to contact us directly by email to try and organize that. And yeah, we'd love to have you in the IMB.
Wonderful answer, Larissa. Just for Charlotte and everybody else's confidence, we will be sending out a bit of a synopsis of what was covered today and a thank you email on the back of that. And so if you have any further questions, just like the one that Charlotte's put forward, revert to that email and we'll put you in touch with the very best person to help you with the next step. Mark, it looks like there's a lot of interest around the piece that you're doing and understanding the viability of once the genes are shed and in the waterways, the lifetime of being able to make your assessment. So one question here is, what is the viability of virions within the wastewater system?
Yeah, so there have been a few studies that seem to indicate they can survive for maybe a day or two, so not very long lived, and a lot of studies that are showing that, in wastewater, they're not detecting live a lot of live virus, but potentially it can survive for a little bit of time at least. But certainly once it's been treated, once particularly in chlorinated water, it dies very quickly.
And I guess the other part of that question also put forward was how many people in community or individuals need to be shedding to find the viral fragments?
Yeah, so the current technology that's detecting the fragmented nucleic acids seems to have a sensitivity around a couple of people per 100,000. So it is pretty sensitive, and certainly within a large suburb can easily pick up 1 or 2 people within that suburb.
Oh, wonderful. And we have a question here also from Neil Smith. And I'm assuming, Neil, that this was off the back of what Loic was sharing with us. How do these genetic risk factors tie in with the non-genetic factors such as obesity, diabetes, etc.?
Yeah, that's an excellent question. Thanks, Neil. At the moment, what we see is that most of the severe forms are observed in people with, in patients with comorbidities such as type 2 diabetes, as you mentioned, and obesity. It's based on our findings. It's still not clear whether there is an actual interaction with the polymorphism that we identify, which are essentially related to the immune response, whether there is an interaction with the other genetic factors that are creating susceptibility to obesity or type 2 diabetes. But this is, I think it's probably because we're still in the early days. As I mentioned, we have about 30,000 cases of COVID-19 in our studies, but most of them are mild cases, so we'll probably need to have larger samples to explore that. So there are some possible potential signals, but I probably won't make too much of it now. But I'll invite you to just look at what we post on this COVID-19 Health Genetics Initiative. It's very it changed quite fast. I think every month we post new results there. So we share everything that we learn. We share it right away. So yeah, watch that space.
Thank you, Loic. And a similar question, but I do believe this was more for Ian. Have you heard that people with little neanderthal in their genes are more susceptible to COVID-19?
Do you know what? I think
for
that's
those of us that are not sporting the PhD after our names, what is that, Ian?
Neanderthals. Those are ancient man. Loic, you probably be able to answer that question much better than I can.
Well, I'm not sure if I will do a better job, but I can speak to that. Yeah, so it's quite interesting. There is so one of the early signals that we identify in terms of the genetic polymorphism was a variant on a part of the DNA sequence, which we now know by looking at how sort of humans evolved, and we can trace back that segments where this variant is from Neanderthal DNA. So interesting that we can see that variance is overrepresented in certain parts of the world, in particular among people with South Asian ancestry and European ancestries. In comparison, the frequency of that variant is quite small in other parts of the world, like in East Asia or Africa. And so that variant seems to be traced back to this Neanderthal, a few of Neanderthal individuals identified. But exactly what is the story there? It's still debated. I think it could be, there are a few speculations there. It could be that there's Neanderthal individuals. Sorry, don't know how to call them. I don't have names, but they're Neanderthal people. Might have faced a similar virus in their early environment, and because there's some actually evidence of selection, positive selection for that variant. So so the evidence is, just to make a long story short, the evidence is that we identify a risk variant which is associated with severe forms of COVID-19. That variant can be traced back to Neanderthal DNA. So there's been some mixing between sapiens and Neanderthal at some point, in particular a part of the world that are around South Asia and Europe. And what exactly that story is, it's still to be explored. But yeah, it's a great paper.
Wonderful, thank you. I can see there's still a few more questions there to touch on in the Q&A, but one thing I'd love to hear from each of you now, and maybe we'll start with you, Ian. In light of the subject matter, how in 2020 has COVID-19 impacted your research space?
Well, we have been fortunate that we've been able to do research, unlike many parts of the world. So, that has been great. But I mean, the really big impacts are probably through funding, where government have redirected certain funding streams, away from the classical support schemes for research. And that has a real follow-on impact onto the continued delivery of research within the building.
Mark, how about you? How have you seen this impact your space?
Yeah, I mean, I'd reiterate Ian's comments about the redirection of funding. Antibacterial funding is generally underfunded to begin with, and in terms of directing funding towards infectious diseases, now there's been the huge focus on, rightfully so, on SARS-CoV-2 and COVID-19 research. But it does mean that the opportunities for getting funding for the antibacterial research that is the core of what we're doing potentially is reduced, even though the threat of antimicrobial resistance is a slow-moving, inevitable pandemic unless we start doing something about it now. I think the other area that has been impacted is, again, as Ian alludes, we've been very fortunate to pretty much keep our research going unhindered within IMB at UQ. But we have a lot of global collaborations for advancing some of the research programs that we're doing, and our collaborators, particularly in the US and Europe, are very much hindered in their ability to do any hands-on experiments. And so that has led to a lot of delays in some of our programs.
Larissa, yourself.
For me, it's been a crazy year, but where I think the toughest bit has been there is that squeeze because of lack of funding, particularly researchers in my career stage. So that's the early to mid-career researchers. Just as funding dries up, despite what we're doing, we kind of get squeezed out. And I think it was Paul Young actually who said that we're probably going to lose a generation of scientists due to funding cuts. And for someone like me, that's pretty devastating. Of course, for me, it's been a great time to be a viral immunologist. I've done more public engagement now than ever before in my life. And that's really exciting to get to be able to explain how things work and why it's important. And it's also, it's pretty meaningful to be able to get in there and work with the virus and understand how things work. But it is a huge demand currently. There are so many projects that we want to do and we can do and we should do, but I'm just one person at the moment, and it'd be great if we could have a few more people in PC3 as well, but currently just space-wise, that there isn't that room. So when ours is up and running, it's going to be fantastic.
Fantastic. And Loic, how about yourself?
Well, I think I'll align with what my colleagues just said. I think funding is probably the number one impact. I will add something I found a bit positive though is the way the community as organized, was self-organized to respond to that crisis. I think that was quite inspiring. I've been quite advocate of open access and things, sharing research broadly in the past, and just looking at how that is implemented in a time when everyone in the world has to worry about their own safety and health, and still pushing so much the research. So that was quite inspiring, and so yeah, I think that was a good point, if I may say, of this experience.
And I think that obviously alludes to the reason that we wanted to share this story today with the broader community, peers in the science sector as well. It's a tough space in science already, and the whole space in terms of funding has been completely changed this year as the government has had to galvanise their efforts around fighting the global pandemic. So I think each of you should be commended on your agility and taking your teams and your colleagues with you on this journey. It has certainly been a very tough year, and we're very grateful for leaning in and looking for solutions in the face of that. There are a couple more questions that I would love to tap in. I want to make sure some people are covered off on. There's a question here. It's come in as anonymous, but I understand that this happens with different strains of certain mosquito-borne viruses. I'm wondering if that's an extension of what you were talking about, Ian?
It is. So Larissa, can I pop that to you as the virologist?
Sure. So are we talking about antibody-dependent enhancement of infection? Yeah, that's definitely the case. So we know that the best with dengue virus. So dengue virus has 4 main strains, and we call them serotypes, again, based on kind of the immune response they elicit. And we know that if you're infected with one serotype and you make an antibody response to it, as you would expect, that those antibodies bind really well to that particular serotype. So if you're infected with dengue 1, you make dengue 1 antibodies. If you get infected with dengue 1 again, you're fine, you're totally protected. But if you get infected with another subtype of dengue, so like dengue 2, 3, or 4, then actually those antibodies can still bind, but they bind really quite weakly. And instead of neutralizing the virus, so stopping it from being able to get into our cells and to replicate, instead, those antibodies sit there bound weakly to the virus, unable to stop the virus from getting in, but actually macrophages, which are my favorite cell type, one of their main jobs is to scavenge up any antibody-bound virus. And what dengue virus does is it uses that as a way to get inside the macrophage and to start replicating. So what those antibodies do, instead of blocking viral infection, is they actually enhance it in cells like macrophages. And we know that that's actually one of the main reasons that people, and particularly children, get really, really sick with dengue virus infections. So because of a lot of study on the mechanisms of how that happens, it means that they've actually understand a bit better what kind of antibodies those are. And so when we're developing monoclonal antibodies against dengue, but also new dengue virus vaccines, so there have been some unsuccessful ones in the past, but they're able to develop them so that we can avoid that. So that's something that we want to really keep an eye on. With COVID-19, it's been shown in cell culture that antibody-dependent enhanced uptake into macrophages can occur. Whether it leads to enhanced infection, we're still not 100% sure, and whether it leads to enhanced disease, we're also not 100% sure. But we get rare reports of reinfection with COVID. Sometimes during the reinfection, people are getting less sick. Sometimes they're getting more sick. We don't know the cause of that yet. So with all the new vaccines being rolled out, with potential reinfection, and that's particularly in places where COVID's running rampant, we really have to keep an eye on what the antibodies are doing. We have to make sure that antibody-dependent enhancement of disease is not occurring in COVID.
I think your passion in this space is undeniable, Larissa, and it's heartwarming. Mark, another question for you. Will you be accepting extracts from plants or bacteria for the antiviral testing, or would you only be accepting pure compounds?
Yeah, so traditionally co-eds operate where we're only accepting purified compounds, and that's mainly because there's such a history of extracts not being able to isolate the active component of it, and it's a lot of additional work trying to isolate the active fraction. So in certain instances, we will collaborate working with extracts, but our preference definitely is for defined chemical structures.
Yeah. And Saeed's put a question in the Q&A. People exposed to different viruses along with COVID-19 at the same time are at higher risks, aka the dengue conversation that we've been having. How would a treatment plan to recover, whether it be the immune vaccines or the vampire therapy, and the success rate?
Oh, that's a good question, but it's probably a little unquantifiable at the moment. Certainly with our vampire treatment, for the 3 patients that we have treated, we've 100% success rate, but that's a very small study, and those people had no other treatment options. So the vaccine stuff that's got, depending, you pick up the newspaper, it seems to be a competition between the two vaccine reporting companies. It's got 90, 94, 95% efficacy. Haven't seen the data, so it's hard to call yet. Larissa, you probably want to comment on that too.
On the vaccines, yeah, I think we'll wait and see. I actually just saw that apparently Pfizer's is now 95% effective as well.
I saw that update this morning in the New York Times.
But yeah, it really depends on the number of cases that they report on. So when they're doing their trials somewhere like the US, where people are exposed to a lot of COVID, it's no wonder that the case numbers rise quite dramatically, and that can have a big effect on sort of the number outcome and how they determine efficacy.
Okay. Still for you, Larissa, do we have adequate in vitro models for the study of SARS-CoV-2 infection at different bodily tissues? And what are our current limitations in the in vitro study of the pathogenesis of this virus?
Yeah, they are great questions. And whether we have adequate models or not, it's tough to answer. In a short answer, no. And that's what we're really trying to work on. So we're trying to study SARS-CoV-2 infection in the context of the lung. So we use lung epithelial cells, and we use human monocyte-derived macrophages. We use primary cells where we can, because we think that if you have an immortalized cell line, that introduces more complications. But
what do you mean by an immortalized cell line?
So, I mean, a cell line that has basically got a cancer mutation, meaning that it continues to grow and replicate, which is really useful for working with in the lab, but it can make it hard to extrapolate biological function from. So we're trying to do our work in the most relevant system. So even though we're working in vitro, we're still trying to look at lung epithelial cell lines as the main target of viral infection. But a lot of people are using this particular cell line called Vero cells, and they're really good at growing virus, but they are a monkey kidney cell line. So we'll still have to see for screening, but it's an ongoing question that we're still trying to establish. And we try and work in sort of co-culture and triple co-culture models, particularly with their endothelial work. And we'd be looking to go into organoid models in the future. But there are, it'd be stuff that'd be great to do in vivo, so in animal models, but we're limited because the virus doesn't infect mice. You need either a mouse adapted version of the virus, or you need mice that express the humanized receptor. So we don't have that yet. And also those experiments are not trivial, considering they have to be done in PC3. So potentially in the future, but certainly not yet.
So to that question, because it sounds incredibly important piece of work, what would make that a reality for you in 2021?
To be able to do in vivo work, so that requires getting the virus in and of itself, which is expensive. Working with the virus, even when we have the PC3 facilities, as I think you saw from my photo, we need to have a lot of personalized protective equipment. So that's all disposable. That's also not cheap. We need specialized equipment for working with the virus, and all of that has to go into the lab. So currently, if we're building the kitchen, we still need to fit it out with the pots and pans and all the ingredients to actually, make that final meal. So that's really kind of limiting us as well. And it's people on the ground. So we need people to help us do that work, and you've got to be quite highly trained to do it. So, most of the people I think all of the people who are currently PC3 trained at least have a PhD. So we're not taking any students at this moment because it is so serious, and we need the highest level of training.
Thank you. Mark, can I put a similar question to you? What is your main ambition in 2021, and what do you need to get there?
Sorry, just trying to get the mute button. Look, in terms of the COVID research, I think getting Larissa's lab off the ground and getting an assay in particular set up. I think the opportunity to screen compounds and build collaborations around the world to screen compounds is very powerful to build on what we've already established with COAD. But that does require a fairly high throughput assay to be developed by Larissa, which is a lot of work. And given the number of different responsibilities she has, I think getting additional personnel in to help with the projects would be very critical.
Yeah. It's really that case of if it's a global challenge, as many hands on deck as possible. Definitely, yeah. Ian, a similar question for you. What is it that you want to achieve in 2021 and what do you need to get there?
In terms of COVID work, Mark, then it's really about actually asking the question, do antibodies play a role in enhancing disease? That's a partnership with Larissa again and with Tim Wells over at UQDI. And to be honest, they'll be doing the heavy lifting in that space, but it's about getting the resources, both the patient resources, because we actually need samples for patients to actually drive that research forward, but also the hands in the lab, because Larissa, is amazing, but can't do everything. So it's about getting additional hands to help her out. But the other thing is really trying to actually see if the vaccine platform that we could build to make vaccines much more rapidly, if that would work. Okay, and that again is work that we're doing with Larissa, but we're probably driven a lot more from my side.
So the point that you made around needing greater samples, is that integrated with the piece that Loic highlighted before? Loic, what do you need next year? How do we help you?
Well, thank you. Thanks for the segue. That was perfect. Yeah, so I think the first effort for us next year will be to trying to get this study going. So this study has a focus on collecting samples from people who've been infected and get their DNA and understand susceptibility, long-term effects. But I think it has to be integrated with the work that Mark, Ian, and Larissa are doing. Having those samples in institute is, extracting DNA is one thing, but there's so much we can do with those samples, and I think that's our focus on the beginning of next year. I think I'll also emphasize the importance of having being able to train students at the high level. I think so one of the things I've been able to do, and because we've been fortunate here in Australia not to have too many cases, so meaning that we couldn't initially contribute samples to that international effort, but because of our unique expertise in statistics and genetics and data modeling, we've been able to make strong contribution to those consortia, and I think that's one of the unique strengths we can continue to build on and train new scientists with us here within the Institute.
Wonderful. Thank you so much for that, Loic. I wanted to take this opportunity to thank all four of you for your generosity of time and input today. I think it's been a wonderful illustration of the sheer excellence of scientific investigation we have underway here at UQ's Institute for Molecular Bioscience. And it is in safe hands with these incredible researchers. I think what we have touched on throughout today's presentation is the symbiotic relationship between what they are doing and some of the ways that valuable support from community members has actually enabled this research. And I just want to take this moment to actually highlight that we have a couple of Director Circle members on the line today, and wanted to say hello to Del directly and also Shane. We're very grateful for you partnering us in our initiatives. If you're interested to know more about the Director Circle, it is a collection of like-minded people who share a passion for discovery with our researchers and are giving at a set level of support to IMB to help enable our research. Given what we've also shared with you today, there are some significant priorities we have in a fundraising space, such as capacity building investment into the Centre for Inflammation Disease Research and the Centre for Superbugs, support to people just like Larissa, who are at the nexus of that generation of researchers that are in jeopardy despite being tremendously talented. and also that those kind of capacity building support roles so that we can get on and get this research energized the way that it should be. But today primarily was about sharing with you how we have galvanized as an institute in collaboration with our peers across the UQ landscape to really make a dynamic difference for the broader community at large, which has been the mission of this institute for the past 20 years, and it is not changing in any way. So with that, thank you very much to everybody for joining us today. As I mentioned, we will be following up with you on the back of this presentation. If we didn't get to your questions today, please reach out to us directly and we will be sure to put you in touch with the very best avenue to help you with that. And if you are doing nothing else on the back of joining us today, we hope that you go and share this message and share these incredible areas of research with at least 2 or 3 other people around the office or the home or within your loved ones or your family group, because it is incredibly important, the work that is underway. Thank you very much to our panelists. And please all, given that this is our last of this series for the year, this is my final opportunity to tell you to have a safe and connected Christmas with your loved ones. And thank you for your ongoing support to this series throughout the year. Take care, everyone.
Thank you, Kamara. Thanks, Marlon. Thanks all.
Bye.